Alright guys, in today's lesson we are going to continue talking about separating mixtures, but we're going to learn a few different procedures other than distillation today. There is a bookwork associated with this lesson, so start your classwork page, which has this homework assignment at the top. And underneath that there's an aim, which is how can we separate particle mixtures using decantation, filtration, separatory funnels, and chromatography. And then underneath that, please copy down these do now questions. Okay, so I have a short video for you to watch about paper and thin layer chromatography.
In this lesson, we will learn about paper and thin layer chromatography, both of which are used in forensic science applications. Chromatography is a separation technique, and this will be demonstrated using food coloring. Using a pencil, draw a line at the bottom of a strip of chromatography paper. Dab a small spot of red food coloring on the pencil line and repeat with green and blue food coloring. Place this in a beaker with water, ensuring that the water level does not touch the spots. As the water is absorbed, it separates the multiple components of the food coloring. We have now created a chromatogram. The chromatography paper is called the stationary phase as it stays put. Water is called the mobile phase as it moves up the chromatography paper. Here is the key idea. A component that is more soluble in water travels a greater distance up the chromatography paper.
Alright, so the first one to talk about today is chromatography. You might notice in the picture to the left that you are seeing some ink or some pigment which is traveling up a piece of paper. You probably did either a simulation of this or a real version of this in your living environment class last year, where you put a sort of homogeneous looking ink in the bottom of the on the bottom of the paper. Perhaps it was red ink in this case. And you would think that the ink is only red, but as it travels up the paper, you'll notice that there are other colors in the ink, such as this blue, and that blue ink was in the red ink, and it starts to separate from the red ink based on their different attractions for the paper. So in my explanations up here, I did mention that this is evidence that the ink is a mixture rather than a pure substance, and in my last question I asked, why would chromatography be useless on pure substances? And the reason is because chromatography is only going to be able to create an observable pattern if the molecules in the substance have differing properties. But a pure substance would contain molecules that all have the same properties, and therefore wouldn't be separable. Okay, so filtration is another really easy one to understand, which is a way to separate mixtures. You can see in the diagram to the left that we have two substances in a mixture, some yellow balls and some red balls, and they're trying to travel through a membrane that has pores in it, but only the yellow balls are able to get through, and that's based on their size. So one thing that we need to know about filters is that they separate particles based on size, and they would never ever work on a homogeneous mixture. Homogeneous mixtures have particles that are far too evenly distributed to be separated based on their sizes. So I included some notes based on the things I just said. Yellow's being separated from red. They're being separated based on size. Heterogeneous mixtures are perfect candidates for filtration, like sand and water, and then homogeneous mixtures are terrible candidates for filtration. They cannot be filtered. So like sugar and water or salt and water, neither one of those things could be filtered. And it's because in a homogeneous mixture, the particles are too evenly distributed to even be separated based on size. So one of the oldest tricks in the book for separating mixtures is called decantation, but that's kind of a fancy term for something that many of us do all the time. So this handsome fellow right here is doing something called panning for gold, which is a form of decantation, where he's separating solids and liquids that are not soluble in each other. So he literally is just pouring the liquid off the top of the solid. Um, and you guys do this actually all the time in your homes, because anytime you have to strain a liquid from a solid, such as when you're straining the water out of pasta, or if you're straining the liquid from maybe some blueberries or something, then that would be called decantation if you were in a chemistry lab. So the key things to remember here are that decantation would only be possible if the solid is not soluble in the liquid. So therefore, any solid which is soluble in a liquid, it couldn't be decanted. So I chose an example like that with salt and water. You wouldn't be able to decant a salt water solution. And the last method that we're going to learn about today is using a separatory funnel. It's a really cool just piece of glassware. It has this valve down here which controls the flow of the liquids, and you basically put two liquids in it, and they should be two liquids that are not miscible, meaning they don't mix in any ratio. Examples could be oil and water. And since oil and water don't mix, and also because they have different densities, the oil will actually float above the water. And we can rely on this property that they have for each other to separate them, because we would simply open the valve right here, and the water would drain through the funnel. And when the border between oil and water, as it approaches the tip of the valve, we would turn the valve off and stop the flow. And what you would have done there is separated the water, which would be down here in the whatever flask you have, and the oil would still be in the separatory funnel. So that's one way to separate mixtures, as long as it's two liquids that are immiscible, meaning they do not mix in any ratio. Examples, oil and water. So I basically just wrote in that two immiscible liquids are being separated, and we do need them to have different densities, and that will allow the separation. And two examples of things that could or could not be separated using a separatory funnel could be oil and water would work because they're immiscible, and alcohol and water would not work because they're miscible. And you've reached the pair up. So underneath the homework assignment and the aim and the do now, please copy down these pair up questions for your separate classwork page. And underneath that, please copy down these summary questions. I know there's a bit of a formatting issue here, but this is question 1, this is question 2, 3, and 4, and these are your 4 answer choices for each question. So go ahead and copy those down, and we'll have them ready in class tomorrow. Thanks for watching.